Hi, my name is Sarah Meeks, and I'm the Vice President of Business Development at Maxcyte. Thank you to the organizers for this virtual cell and gene meeting on the med. The title of my talk today is Partnering with Maxcyte, Driving the Next Generation of Cell-Based Therapies. So who are we at Maxcyte? We're a collaborative, innovative, and experienced partner. Our flow of electroporation technology was invented at the CBR Biomedical Research Institute in Boston. The company was founded in 1999 to commercialize this technology. In 2000, we developed the Maxcyte GT scalable transfection system. And in 2003, we began to enter license agreements with cell therapy developers. In April 2019, we launched the Expert technology platform, which represents the next generation of the industry's leading clinically validated flow electroporation technology for complex cellular engineering. To date, we have 12 commercial partnerships, over 100 clinical program licenses, and over 140 total licenses, including our clinical and commercial, as well as research and academic licenses. We continue to rapidly expand our cell therapy partnerships with the leading industry and academic innovators. So today I'd like to tell you a little bit about partnering with Maxcyte. My role at Maxcyte is to lead up licensing of our flow electroporation technology to cell therapy developers in the sector. Presently, there are many challenges with cell therapy engineering that still remain. The choice between viral and non-viral. If a virus is the choice, it's whether that is available, if you can schedule manufacturing, the costs, and there are safety issues remaining. Technology choice is still a challenge. If the technology from at the front end of R&D is incompatible with the back end, you may need to re-optimize, you may need to revalidate. The efficiency and scalability of that technology can be a challenge. There's plenty of competition in the marketplace now. There are many companies and organizations entering into the sector focused on the same indication, and speed to market is critical. And finally, costs can be a challenge. Lack of efficiency in that translation throughout the product development cycle, virus supply, all of these may delay translation, of which technology choice will influence your timelines, your cash burn. It could lead to increased risk and higher COGS, which in turn affect your speed to market, the affordability of your product, and access to that product. But innovation is driving improvements and new opportunities. There are an increasing variety of cell types being engineered, such as Tregs, NKs, gamma deltas, myeloid cells. There's continued expansion of cell sources, i.e. iPSC or INK, cord blood, placentally derived, and improved therapeutic approaches, autologous improvements, allogeneic improvements, batch, large-scale batch, neoantigen approach, and increasingly complex engineering for novel product functionality, i.e. multiplexing, novel knock-ins or knockouts, or combinations thereof. And finally, an ever-widening field of innovative technical engineering and editing platforms are being employed. So at Maxcyte, as we developed our expert system, prior to that, we went to the market and asked a couple of critical questions. What first was the critical top of mind improvements desired in a technology that would provide cellular engineering? And the three top of mind issues that emerged were efficiency, viability, and reproducibility, with efficiency being a critical need. This survey was done from 100 non-Maxcyte customers. We also asked that group how important it is to choose a single transfection technology that can meet your needs from early research into late stage preclinical or even into the clinic. And on a scale of 1 to 5, 1 being not important and 5 being very important, the importance of a single transfection technology was critical, i.e. scalability is highly important. And again, this is a survey that was done with non-Maxcyte customers. So from there emerged a vision of one unifying technology from concept to commercialization. This single technology platform would enable cell type versatility engineering complexity, had to be highly efficient through scale, preserving cell viability and potency. There need to be reproducible, and of course it needed to be scalable. Overall, enabling the opportunity for significant reduction in timelines, risk, and overall cost. So in 2019, the Expert platform was launched. And this is a platform, a technology platform that's comprised of an instrument family of high performance single technology platform instruments, from a research grade ATX instrument through to our clinical grade GTX instrument, paired with a set of consumables that will accommodate research studies and scale seamlessly to clinical and commercial manufacturing. Overall, significantly reducing time and resources by eliminating the need for re-optimization and/or revalidation. A little bit more on the instruments. This is a diagram of the instruments and the features of the set of instruments in the family, the ATX being across the top here. This is our research grade instrument, and this is our static electroporator for used in R&D. At the bottom here is our GTX instrument, or our GMP compliant instrument, which is our flow electroporator and includes, it's 21 CFR enabled. There's access to the master file with this instrument, and it can be used in clinical and commercial manufacturing. It's the same platform on all of 3 of these instruments though. So there is no need to re-optimize or revalidate in moving from R&D into clinical scale and eventually into commercial scale. A little bit on the expert processing assemblies, PAs, as we call them at Maxcyte, are our consumables. And these are a set of scalable high performance transfection consumables. And they've been designed with both R&D in mind, as well as through translation and to clinical scale for a variety of different types of applications and clinical cell therapy approaches. At the research end, for example, our OC 25x3, the minimum volume is 120, or excuse me, cell count as 125,000 up to 5 million, up to our CL2 to be used in our flow electroporator GTX, from 500 million cells to 20 billion cells can be accommodated, or up to 100 mils. The 2 instruments across the bottom here show the PAs that are comparable or can be used with each of the instruments. We have a number of new PAs that have been introduced or in the process of being introduced. These are the new product focuses are on a range of sizes, improvements to handling, and they're going to be both research as well as clinical grade PAs. New for the second half of 2021 is a multi-well research PA and a mid-scale flow electroporation for clinical use. But at Maxcyte, Expert isn't just our technology or our processing assemblies, it's a partnership. And our goals as partners with you are to align on creating value and enabling speed to market for your product. It really falls into 4 main categories. I've talked about the instrument. Under that would be a full warranty, high performance GMP 21 CFR Part 11 equipped and network compatible instrument. As I described, this is our GTX here that I'm describing, but also the ATX is the research instrument that many organizations start with in R&D. With that partnership comes technical support, and this is there from the point of conceiving your product all the way through commercialization. Access to our team of field application scientists is throughout process development, translation, clinical development. There's a deep and varied technical set of expertises on this FAS team. It's full global support, i.e. enabling both expansion up of your product as well as manufacturing out. They're experts in optimization and process development and are there for ongoing problem solving, again, from concept to commercialization. There's quality systems, including GMP manufacturing, ISO 9001, safety stock, and finally, regulatory support, which would include access to our FDA master file as you file IND, or in other countries, the equivalent of the IND. We've got a regulatory support team that is going to work with you wherever that clinical trial is going to be conducted. And there's geographic support, again, as you not only scale up, but you scale out. A little bit more on our technical support. We've spent a great deal of time and effort building out this team very specifically to support and to gain scientific expertise to support our partnerships. And our field applications scientists have expertise in immunology, gene editing, iPSC and stem cells, viral vectors and vaccine cell-based assays, and process development. And they're located globally with a total of a cumulative experience of over 350 years experience. In addition, our partners are also enabled to access our R&D team at headquarters to help with problem solving, optimization, process development. And again, as you scale up and scale out, this technical team, field application specialists, will move with you as your programs expand. So if there's an expert that worked with you in one location early on, that expert's gonna go and do tech transfer for you, even if that technology is being transferred across the globe. What's our progress to date? So we have 12 commercial partnerships and a number of academic and research partnerships. We've enabled a number of cell therapies in clinical trials already. This list is not completely inclusive, but it highlights some of the main areas in genetic diseases and solid tumors, infectious diseases, the heme malignancies, and a number of others. And these programs are either presently in the clinic or have been in the clinic in the past. This list continues to grow. And we've also become the partner of choice for complex cellular engineering. This slide represents a number of our clinical and commercial partners, as well as some of our key academic partnerships. And again, we have 12 commercial licenses, over 100 clinical program licenses, and over 140 total licenses, which is going to include those academic licenses and our research licenses. As you can see, there are a number of different cell types that are being enabled here, as well as editing and engineering modalities, companies focused on different diseases, and this continues to grow. Finally, Maxcyte strives itself on a collaborative partnership from concept to clinic. We want to be your cell therapy partner. We have a high-performance electroporation platform that's efficient, preserves cell viability, it's reproducible, and it's scalable for virtually any cell, any cargo, and at any scale. Our FAS team has an enormous amount of support and will be there for product development. Our access to our R&D team is included, quality and alliance management. There's access to our regulatory team, including regulatory certifications, our drug master file. We have a broad IP portfolio, which is providing enhanced protection and freedom to operate for our partners. There's a seamless and efficient transition from research to commercial scale on one platform, the expert platform, the family of instruments, as well as scalable consumables. And finally, a partnership with Maxcyte means working together to accelerate your path to the clinic, to help patients and to save lives. If you're interested in learning more about a partnership with Maxcyte, please contact me. My email is below. Thank you.